Okay, good morning, everyone, and welcome back from Thanksgiving. Hope you had a good break. A great break. Okay. Okay, I think most people are here. Let's see. Okay, I'll wait 2 more minutes for others to join. Okay, well, let's get started. So today we're moving on to chapter 16, which is on the gene regulation in eukaryotes. Last time we spoke, we looked at regulation in prokaryotes. And the slides for today should be posted. They are posted, so it would be helpful if you open those and follow along. I will show my screen, but I know there have been glitches in the past, so be sure to follow along with the slides from the Learning Hub that I posted on the Learning Hub. Okay, so let me share my screen with you. Okay. Just a sec here. Things are a little crazy. I'm looking for... I can't find the Facebook, the Facebook page. Oh my, I think I closed it by mistake. Let me see. Yeah, here it is. Okay, all right. Okay, so I'm going to share, you should be seeing my screen. So let's go from there. Alrighty. Okay, so we were looking at, like I said, prokaryotic gene regulation in prokaryotes. This week we'll look at regulation in eukaryotes, and similar to prokaryotes, gene regulation can occur at either transcription, any of the steps in transcription, and any of the steps in translation, and post-transcription or post-translation expression regulation can also occur. Some factors that add some complexity, however, to regulation in eukaryotes are, for example, chromatin structure. In an earlier chapter, which I believe you covered with Dr. Lyons, the DNA in eukaryotes is associated with protein, and those proteins are of different types. The one that I think was emphasized with Dr. Lyons was histones. So histones abound with DNA in eukaryotes, forming what is known as chromatin. And that chromatin structure plays a role in DNA expression, in gene expression. When DNA is tightly wound with these histones, when the chromatin is tightly bound to, when the chromatin structure is tightly wound up or tightly bound up, that's when the DNA is tightly bound to the histones. It can actually affect the availability of promoters to the RNA polymerase, and that can affect gene expression. So chromatin structure plays a role in gene expression. It determines how available that DNA is for transcription to occur. Another is RNA processing. RNA processing only occurs in eukaryotes, not in prokaryotes. And so regulation is impacted, gene regulation is impacted at that stage of transcription, that is the RNA processing stage. Transcription and translation are uncoupled in eukaryotes. If you recall, when you looked at regulation in prokaryotes, you saw that regulation of transcription and translation can take place simultaneously. So whether or not a particular RNA transcript is further translated, that decision could be made even before transcription ends, because we know that in prokaryotes, there's no separation. The transcript, the RNA transcript is available for the translation machinery because there's no nucleus present in prokaryotes. And as a result, transcription and translation occur together in prokaryotes. But in eukaryotes, that's not the case. We know transcription occurs in the nucleus, and the transcript is then exported to the cytoplasm where translation will occur. And that sets up an additional complexity of of regulating, or I would say another step, another level at which gene expression could be regulated. And the regulation of genes, it says, I'll just read it here. It says gene regulation controls cellular differentiation and determines the hundreds of specialized cells that are found in a typical eukaryote. Most eukaryotes are multicellular, and these cells are specialized. You have specialized cells. So gene regulation controls these various cell types that you find in eukaryotes, and what is expressed from these cell types is determined by gene regulation. So, for example, we have cells found, let's say, found in a pancreas. So, a pancreas produces cells that contain cells, I should say, that produce insulin, and so the expression of the insulin gene in those cells is determined by transcription factors that might be found in that cell, versus a cell that is not producing insulin may lack those particular transcription factors. So gene regulation helps to control cellular differentiation also, and what is expressed from these different cells. So there are many steps involved in eukaryotic gene expression, and let's see some of these. Transcription and initiation. So genes can be regulated at the level of initiation of transcription. It can be regulated at the processing level, which I alluded to before. That's the mRNA processing level. Also, the export from the nucleus, because we know that transcription occurs in the nucleus, translation occurs in the cytoplasm. So the rate at which these transcripts are exported to the nucleus can affect the expression of the gene, and whether or not the transcript gets exported to the cytoplasm affects expression of the gene. So export to the nucleus is another level, another area at which regulation can occur. Translation of the mRNA, whether or not the mRNA is translated, you may have transcript and being produced, mRNA being produced, but it may not be translated. It may be degraded very rapidly and not get to the point of translation. And so that's another level of regulation. Protein localization, where the protein is located in the cell, where it's where it's sent to. If it's sent to the membrane, then it becomes a membrane protein. If it goes into the lysosome, it has a different function. So where the protein is located in the cell and in which cell types also affects expression. And modification. Once the protein is made, it can be modified by adding various active groups. For example, phosphate groups could be added to the protein, and that can affect how that protein functions. Okay, so let's go a little deeper, and we would look at the different types of polymerases that are found in eukaryotes. So in prokaryotes, there was one type of polymerase present that transcribed all genes, but in eukaryotes, that's different. There are 3 polymerases present. There's polymerase 1, which transcribes the ribosome RNA genes. Polymerase 3, which transcribes tRNA genes and small regulatory RNAs. But in this chapter, we're going to focus on RNA polymerase 2, which transcribes mRNAs, messenger RNAs, and also microRNAs. And we learned earlier that microRNAs play a role in gene regulation, regulation of gene expression. So know that eukaryotes contain more than one type of polymerase. Now, when we looked at prokaryotic gene expression, we discussed that there are different types of regulatory elements. We have what are called cis-acting factors and trans-acting factors. Can anyone tell me the difference between a cis-acting factor and a trans-acting factor? Okay, Olivia, it says cis is on the same side. On the same side of what? On the same side of what? You're mentioning it in terms of organic chemistry, but what do you mean by on the same side versus trans? Okay, here's the question. What is the difference? Oh, let's see. What are trans-acting factors and what are cis-acting factors? Clementine, if you're hearing this, go ahead and respond too. Okay, let's see. Rayford says transacting factors means that the regulatory factors can diffuse through the cytosol. Great. Whereas cis-acting affect each other based on their linear proximity to the genome. That is excellent. Trans controls expression on cis sequences. Okay. Let's see. Someone else is responding. Okay, let's move on then. Let's move on. Okay, so cis-acting factors, like you mentioned, examples would be your promoter. We talked about that in when we looked at prokaryotic gene regulation. So promoters are example of cis-acting factors. In prokaryotes, you also have operators. Those were cis-acting factors. And so in eukaryotes, your promoter is a cis-acting factor. Promoters the promoters are the regions where RNA polymerase, in this case RNA polymerase 2 bind to promoters. So these are sequences that are usually adjacent to the gene. They're usually found upstream of the gene or 5' of the gene, and that's where RNA polymerase binds. Often have a Tata box, we talked about that. And promoters allow for basal level transcription, meaning low level transcription. So when the RNA polymerase binds to these promoters, transcription will take place, but at a very low level. Okay. In addition to this particular cis-acting factor, in addition to promoters, there's another cis-acting factor present in eukaryotes, and these are called enhancers. And you're seeing that in this figure, enhancers. And we'll move to this slide. Enhancers can be found upstream, that is 5' of the gene, or it can be found 3' of the gene, downstream of the gene. So enhancers are DNA sequences that can be found either upstream or downstream of the gene, and enhancers allow for they augment basal level transcription. They can either increase it to very, very high levels, or it can decrease, repress basal level transcription. So just a second here. Okay, so enhancers modulate the basal level transcription. So what you should know, what's important to know about enhancers is that they can be found upstream or downstream of the gene. They can be found at very, very far distances from the gene. So they could be located thousands of base pairs away from the gene and still regulate that gene. They can be found, the orientation of these these factors, these enhancers, could vary. They could be in a 5 to 3' orientation relative to the promoter, or vice versa. So enhancers are another type of cis-acting regulatory element that you find in eukaryotes. However, enhancers do not, or I should say polymerase do not bind enhancers. Polymerase only binds the promoter. Enhancers, however, are associated with another type of protein called trans-acting factors, and we're going to talk about these trans-acting factors. Okay, so enhancers are bound by trans-acting factors. This slide just shows you how you can identify how enhancers are identified, because enhancers are DNA sequences. And let's say as a researcher, you're trying to determine if a particular sequence is an enhancer. We know how to identify promoters, because there are certain sequences, consensus sequences, that you will find in promoters. But you will enhancers don't necessarily have consensus sequences. But how, as a researcher, if you wanted to identify an enhancer, how could you do that? And you could do that by using reporter genes. And we talked about reporter genes in the past. You could take a particular sequence that you suspect that might be an enhancer, and you put it, you could put it upstream or downstream of a particular the promoter of a particular gene, and measure expression using a reporter gene. In this case, in this figure, they're using a reporter gene known as GFP. GFP stands for green fluorescent protein. And when this protein is expressed at high levels, you can see that effect in the phenotype of the organism. So here in this figure, you're seeing here an adult eye specific enhancer, and this is the promoter for that gene, and this is the transcript produced from that gene. Under normal circumstances, if you place a green fluorescent protein downstream of the promoter, you will have some green fluorescent protein produced, but that again will be produced at basal levels. But if you should add your enhancer, or what you suspect to be your enhancer, and it increases expression of that protein, you will have higher levels of or increased levels of the green fluorescent protein. So you can create this, you can take this construct and create a transgenic organism. This is a transgenic organism. We create a transgenic organism if you take this adult eye specific enhancer and promoter from an organism, and you take the DNA from that DNA and ligate it to the green fluorescent protein. Then take this construct and place it into an organism, in this case, mouse. You can create a transgenic organism, and a mouse with expressing the GFP in the eyes. Okay, so that's a nice little green-eyed mouse, right? Okay, so let's move on. So that's how you will, that's one approach to identifying an enhancer sequence. Okay, so in addition to cis-acting elements, which are your promoters and your enhancers, the eukaryotes also have trans-acting elements or trans-acting factors. These are called transcription factors, and trans-acting or transcription factors bind to enhancers. They bind to enhancers, and they can augment gene expression. You also have some transcription factors that can bind promoters, okay? Can also bind promoters, okay? But RNA polymerase never binds, will not bind an enhancer, but transcription factors can bind either promoters or enhancers, okay? And so it says transcription factors are specific DNA binding proteins. So they have the ability to bind DNA, they can bind promoters, they can bind enhancers, and what they do, they recruit other proteins to either the enhancer or the promoter and influence transcription that way. Okay, so transcription factors are also called trans-acting factors. So they're able to diffuse through the cytosol, and they can bind to either enhancer or either promoter, and influence by recruiting, they recruit other proteins to the transcription machinery and influence transcription that way. So what are these transacting factors or these transcription factors? There are three types. One type is known as your basal factors. So basal factors bind to the promoter. Basal factors bind to the promoter and allow for basal level transcription. So in this figure, you're seeing RNA polymerase bound to the promoter, but here are some basal factors. You have what is known as TBP, which stands for Tata binding protein. Tata binding protein binds to the Tata box, okay? And the Tata binding protein, once it bounds, it then recruits the TAFs. TAFs stands for Tata binding protein associated factors, okay? And so TBP binds, that's then followed by the TAFs. And once the TAFs binds, it then recruits RNA polymerase to the promoter site. And this complex allows for basal level transcription. Okay. All right, let me see. Are there any questions so far? Let me check. This is a little awkward here. Okay, Kimberly, you're asking, let's see, cis-acting affect the expression of adjacent. Oh, you're now answering the question. Okay, I get it. Okay. Yes, let's move on. Okay. Okay, so the binding of these basal factors is very ordered. It says this is an ordered pathway of assembly at the promoter. First, you have the Tata binding protein binding to the Tata box. Then that's followed by the TAFs binding to the Tata binding protein. Then that's followed by the RNA polymerase 2 binding to the TAFs. And so the binding occurs in that order. And that complex allows for basal level transcription. Okay. We know that these transacting factors or transcription factors also bind enhancers, and enhancers help to, it says are responsible for varying the levels of transcription. It can cause an increase in transcription or it can cause a decrease in transcription. And in the trans factors or the transacting factors that are bound to the enhancers associate with the basal complex by folding of the DNA. So the DNA bends and folds and allows the complex at the promoter to associate with the complex that is bound to the enhancer. So this is showing you here in this figure the mechanisms of how an activator can affect transcription. So I think I missed something. Let me go back. We were talking about the transacting factors that bind to the enhancers and promoters. So we said enhancers and promoters are cis-acting elements. Then we have our transacting elements. The first one I mentioned was the basal factor. Activators are another transcription factor. So we have our basal factors. Now we have our activators, and activators will bind the enhancer, and that's you're seeing that here. Associate with the promoter, the basal complex that's at the promoter site, and that will lead to an increase in gene expression. So in activators increase gene expression. And this figure is showing you mechanisms by which that's done. So here we have, in this first figure, we have the enhancer associating with the basal factors at the promoter site and increasing expression that way. Or you can have activators can recruit co-activators. So they recruit co-activators, which is shown here, and these co-activators can displace nucleosomes, and the displacement of the nucleosomes cause the chromatin to unwind and make the DNA available for transcription to occur. Okay, because that binding of the DNA with the nucleosomes, those are the the binding of the DNA to form nucleosomes, which you know are complexes of DNA with histones, those inhibit gene expression. So these coactivators, when recruited, they help to remove or disassemble this chromatin structure, disassemble the nucleosomes, and hence make the nucleosomes, make the DNA available for interaction with the transcription factors and allow for expression to occur. Okay, let's look at the physical structure of these activators. So activators, we know, have to be able to bind DNA, and there are two key features that you'll find in these activators. So they have, it says activators have at least two functional domains. They have what is known as a DNA binding domain, and this figure is showing you two of those DNA binding domains. One is known as a helix turn helix. This is referring to the secondary structure of the protein. So remember that activators are proteins, okay? These are transacting factors are proteins, and proteins can form secondary structures. And so the helix turn helix, which is shown here, is a secondary structure that binds into or associates with into the groove of the DNA, the major groove. You're seeing that here, found in the groove of the DNA. So this is the protein, and it binds to the DNA. Another typical DNA binding structure is known as a zinc finger. This is a zinc finger, another secondary structure. And this structure has a lot of bends and loops, but it's associated with the zinc, with a zinc atom. And so it's called zinc fingers. And that is another structure of that allows for a protein to bind to DNA. So activators have these DNA binding domains known as the helix turn helix and the zinc finger. There are others, but the text is only focusing on these two types of DNA binding domains. In addition to DNA binding domains, activators also have what are called activation domains. These activation domains, it says, it binds to other proteins like the basal factors and coactivators. So there's a portion of an activator that binds to the DNA, and there's another portion that binds to other proteins. And these other proteins could be either basal factors or it can be coactivators. Okay, so activators have two functional domains: DNA binding domains, of which we have two examples, and you have activation domains. Okay, some activators also have what is called a dimerization domain, and dimerization domain allow for activators to form what are known as homodimers or heterodimers. So it allows for multimeric proteins to be formed, and these multimeric complexes could be either homodimers. If all the subunits are the same, they're called homodimers. But if they're not identical, they're called heterodimers. So here's an example of a DNA binding protein known as JUN. So this is a homodimer because both subunits are the same, okay? And here's JUN and FUS. This is an example of a heterodimer because the subunits are not identical. But here, as you can see here, this is another feature known as a leucine zipper, which is also characteristic of DNA binding proteins. And you can see here it's bound to the DNA there, okay? So that's an activator. So activators they have DNA binding domains, they have activation domains. Some, not all, but some activators also have a dimerization domain, okay? All right, so far we've talked about two types of transacting factors. We have our basal factors and we have our activators. There's a third type. these are our repressors. Repressors are a third type of trans-acting factor. And we talked about repressors in when we discussed the prokaryotic gene regulation. Okay, so repressors, like what it says, it suppresses transcription, and it does that by recruiting. It could do well. There's several mechanisms that can be used, and one is to recruit co-repressors. Okay, can recruit co-repressors. It says repressor proteins suppress transcription initiation by recruiting co-repressors, and there are two alternate functions of co-repressors, and it's shown you here. So here we have the repressor bound to the enhancer. Here's a co-repressor that is bound to that repressor, and in this case, the co-repressor, this complex, that's the repressor and co-repressor, are preventing RNA polymerase from binding to the promoter site. So that's one way of affecting or suppressing transcription initiation, by preventing the RNA polymerase II complex from binding to the promoter site. Repressors can also act by causing the nucleosomes to form. They can recruit, it says here, recruit co-repressors that close the chromatin structure. They can cause the chromatin to bind very tightly, and once that occurs, then that would inhibit initiation of transcription. Okay. So it says here in the slide, these co-repressors can prevent RNA2 complex from binding to the promoter, or they can modify histones to close the chromatin structure. Here's another way that repressors can act. They can act also via competition with the activated protein. Okay. So it says here, an indirect repressor interface with the function of an activator. So they can compete. They can compete with the activator for the binding site on the enhancer, because we know that both activators and repressors bind to enhancers. So you can have a repressor that will compete with an activator for the enhancer binding site. Another is known as another mechanism is called quenching, where the repressor binds to the activator, the activation domain of the activator. Because remember, activators have a DNA binding domain and an activation domain. The DNA, so in quenching, the activator is able to bind to the enhancer, but the repressor comes and binds to the activation domain. And once the repressor binds to the activation domain, no other protein, no co-activator or no basal factor is able to bind. And that is one way of repressing or suppressing transcription. Okay, so quenching is another. So there's competition, and then there's quenching. Then there is binding to the activator. The repressor could bind to the activator and keep it in the cytoplasm. So it's known as cytoplasmic sequestration. We know that transcription takes place in the nucleus. Once if you could prevent the activator from getting into the nucleus, that is one way of suppressing transcription initiation or enhancing transcription. And so sequestration of cytoplasmic sequestration is another way of regulating, or I should say, another way a repressor protein can affect an activated protein. And we have another known as homodimerization, where the binding to an activator and preventing homodimerization. So you have some activators, like we mentioned, require dimerization for them to function. What a repressor can do, one way of affecting that, a repressor could bind to that one subunit of an activator that requires dimerization to function and prevent that activator from ever forming a homodimer, and and thus inhibiting or suppressing the function of the activator. Okay. Okay, let me see if there are any questions so far. Okay, no questions. Let's see. Okay. Okay, because eukaryotes tend to be, most eukaryotes are quite complex organisms, are multicellular. You can have and contain many genes, usually many genes. You can have the same transcription factor playing different roles in different cells. Okay, so for example, you're showing here in this figure, it's showing you an activator known as dorsal protein in Drosophila. This dorsal protein can act as an activator to one gene and then act as a repressor for another gene. So it's showing you here that dorsal, here in this first part of the figure, dorsal when complex with a coactivator would enhance transcription or increase transcription of gene 1. But that same dorsal can interact with another transcription factor in connection with a co-repressor and act as a repressor for another gene called, well, gene 2. Okay. The point is that the same transcription factor can act as a repressor or an activator depending on the cell type. Okay, it's just an efficient way of controlling gene expression. Instead of having to make too many, you think of it this way, and instead of having a cell, having an organism being required to make lots and lots of transcription factors, you can have one transcription factor having more than one role in the cell. Okay, so if this is another example of how you can identify transcription factors. Earlier we looked at how you can identify enhancer sequences, but if you had a protein and you suspected it was a transcription factor, you can determine if it really is by again using this reporter gene construct. So here, what you can do is to create mutations in the genes that make these in the genes that make these activators or repressor. We know an activator is supposed to increase expression of genes, increase the expression of genes. So if you create a mutation in a gene that encodes an activator, it should cause a reduction in expression of the genes that activator affects. And the same can occur for a repressor. If a mutation in a gene that encodes a repressor can cause an increase in expression of the reporter gene. So here you're seeing an example of that. So we know that here's an enhancer showing it bound to a promoter, and that promoter is bound to the reporter gene, in this case, the green fluorescent protein. And under normal circumstances, that enhancer will cause high levels of green fluorescent protein to be produced. To determine if a particular protein is an activator, you can create a mutation that either prevents that protein from being made in this example. So there's a mutation in this potential activator protein, and that leads to low levels of GFP. So, okay, so you can see here that this fusion construct can be used to identify cis-acting elements, and it can be used to identify trans-acting elements. Okay. All right. So just I mentioned that these eukaryotes tend to be quite complex. They're usually multicellular organisms. And in humans, for example, we know we have about 25,000 genes, and about 2,000 of those 25,000 genes encode transcriptional regulatory proteins. Okay? Each of these regulatory proteins can act on many genes, and one gene can be regulated by many transcription factors. So many proteins can regulate the transcription of one gene. Okay, so it's regulation of, I think the point here is to show that regulation of gene expression in eukaryotes are very complex, because we have lots of genes involved in making transcription factors. These transcription factors can act as activators or repressors. The same transcription factor, I should say, can act as an activator or repressor. And then you have multiple transcription factors affecting one gene, or you can have multiple, many transcription factors affecting one gene. Okay, so gene expression becomes quite complex, but this level, this having such a high amount of transcription factors allows for gene expression to be really very well fine-tuned in eukaryotes. Another thing to bear in mind is that during the development of a eukaryotic organism or multicellular organism, you will have gene expression being turned on and turned off at different phases of development, and that is all controlled by the amount of transcription factors that are produced, the type of transcription factors produced in that particular cell. So for example, you will not want a pancreatic cell, although it has the gene for melanin, for example, you won't want melanin to be expressed in pancreatic cells, okay? Neither will you want insulin to be expressed in skin cells. So in eukaryotes, think of it this way. Eukaryotes, you have all the cells have the same genetic information, but yet the genes are expressed differentially depending on the cell type. Okay, so that helps to add, that adds to the complexity of gene expression that occurs in eukaryotes. And so this figure is showing you here, highlighting that. Here we have two enhancers. It says an eye enhancer here and a skin enhancer. Here's the promoter. And in this example, you're seeing here the activators, which are in green, are not bound to either enhancer, but repressors are bound. And once the repressor is bound, then you have no transcription occurring from that gene. Okay. In the second part of this figure, and I should point out, this is occurring in the G1 phase of the cell cycle. In the S phase, you're seeing here that the activators are bound to the eye enhancer, and that allows for expression to occur. And then you could also have activators bound to the skin enhancer, allowing for expression of that gene that's in the skin that is necessary to be expressed in skin cells occurring. So one way of regulating expression is by which transcription factors are bound to which enhancer. So that's another way of regulating expression. We have one more minute. I don't know if we can get to this. I think we'll end here. What I would like to do, what I will do, there's a video that I'm going to post on the Learning Hub that I would like you to look at in preparation for Wednesday. So on Wednesday, we're going to be talking about epigenetics, and we're looking at factors outside the gene sequence that can affect gene expression. And so there's a pretty neat video that I will put the link up for on the Learning Hub. So I would like you to take a look at that in preparation for Wednesday. So I will see you on Wednesday.